Hello, biologists. We're going to practice for the final exam by taking a pretend quiz. This is going to be 20 questions long, and I want you to treat it as if it was an actual quiz, and we will go through the answers together. I do want you to answer each question for yourself first, so maybe get ready to pause this a lot and then restart it after you've had a moment to talk about it, because I'm going to go through it quickly. Question 1. You analyze the structure of a molecule and determine that it is made of a chain of amino acids. What molecule are you analyzing? A protein. Question 2. Which of these molecules has a complex three-dimensional shape? C. A protein. What are the two copies of a chromosome connected at the centromere called? They are sister chromatids. DNA polymerase III is able to add new nucleotides only to the blank end of an existing strand of nucleotides because that is where the blank group is found. The 3' end, because that's where the phosphate attaches. Which label identifies a 5' end of this DNA molecule? C. The 5' end is up by the phosphate group. It would be right here, outside of the pentagram. What type of bond is shown inside the red box? Hydrogen bonds. Which label is on the leading strand, A, B, or C? A is on the leading strand. What is R identifying? Helicase. It's unwinding the double-stranded DNA, creating the replication fork. N is synthesizing new DNA continuously. What is N? That's DNA polymerase III. Which of these is the least likely to be shown in this picture? DNA polymerase II. DNA polymerase II is involved in gene editing. It's going to look for errors in DNA. So it will not be used during replication. It'll be used after. During DNA replication, what would happen if DNA ligase wasn't functioning correctly? The lagging strand would not be connected, and so DNA replication would occur in unconnected chunks, and it wouldn't be effective. You would have little broken pieces of DNA instead of one long strand of DNA. During DNA replication, what would happen if primase wasn't functioning correctly? Primase lays down the primers. Without primase, DNA helicase III cannot add new nucleotides. That means that DNA would not be replicated at all. The MYBPC3 gene causes hypertrophic cardiomyopathy. It's the leading cause of sudden death among young athletes. What are the subunits that would make up the MYBPC3 gene? A gene is a piece of DNA, so these subunits are nucleotides. Which of the statements below is true about DNA and RNA? Both DNA and RNA contain a 5-carbon sugar. In a DNA molecule that is made of 20% adenine, what percentage would be made of guanine? It's a Chargaff's rule. If I have 20% A's, I know that I have 20% T's. That means that the remaining 60% must be divided between C's and G's. So I'm going to have 30% G's. You have identified a new protein in Drosophila, which is fruit flies. After extensive research, you have determined that the protein is 333 amino acids in length. How long is the protein coding portion of mRNA for this protein? 333 amino acids. means you need that many codons. Each codon is 3 nucleotides, so 999. If the template strand of a DNA molecule has the sequence 5' GTAC GT3', what is the sequence of the non-template coding strand of DNA? They are anti-parallel. So the non-template strand would begin with 3', and then you would complementary base pair C, A, T, G, C, A, 5'. If the template strand of a DNA molecule has the sequence 5' AAA GGA 3', what amino acids would be coded for in the mRNA? So first we're going to have to do transcription. We know that our DNA is anti-parallel, so it'll be 3' to 5'. And then we do complementary base pairing, but mRNA does not use T, instead it uses U. So we have A pairs with U, A pairs with U, A pairs with U, G pairs with C, C, and U. So now we have 2 codons, each of 3 nucleotides. And when we do translation, translation is going to read the mRNA from the 5' to the 3'. So it's going to go in this direction. So we're going to do the 2 mRNA codons backwards. So we're looking for U, C, C. We'll use our chart, first position, U, so it's going to be in this row. And then C, it'll be in this box, UC, all these start with U, and then C. Our last letter is C, so our first amino acid will be SER, that's amino acid number 1. And our second one will be UUU, so same row, but now we're in the first column, and we're looking at this one here. So amino acid number 2 would be Phi. Delete all that. Okay, question number 20. If a DNA template strand had this sequence, 5' ACG, Max is making an appearance, ACG, TGT 3', what would the amino acid sequence be coded for in mRNA from that DNA? We'll do the same thing. We're going to get our pencil. We know that it's anti-parallel, so it'll go 3'. What pairs with A in mRNA? That's U. And then G, C, A, C, A, and 5'. We're then going to go backwards because translation occurs at the 5' first. So we're looking for the codon ACA. That will be in this row, A. Second position, C, so it'll be in this box. And ACA codes for thir. That's going to be our first amino acid. Our second amino acid, CGU, C, column G, so it's going to be in this box. And CGU, our second amino acid, is going to be R. If you have any questions, please feel free to email me anytime. A fun thing to do would be to go through this quiz a couple of times until you feel really confident, and to look at each question and see how you can alter it to ask another question that's really similar. For example, maybe you alter the DNA sequence in question 19 and ask yourself another question, or you go back to our picture of DNA replication and you make sure that you can identify each of those letters. So use this to expand your studying, and email me anytime. I hope you have a great rest of your day. And I want to end on this slide because look at those two. Wonderful. Have a good day. Talk to you soon. Bye.